Let's review the clues to an anaerobic infection. A specimen may suggest anaerobes if there is a foul odor, if the infection is located close to a mucosal surface, or if it originates from an animal or human bite. The presence of gas in the specimen is also suspicious, as is a history of previous treatment with aminoglycosides, which do not cover anaerobes. Other signs include black discoloration in blood-containing exudates, the presence of sulfur granules, or a unique morphology on gram stain. If organisms are visible on the smear but fail to grow aerobically, that is another clue. Finally, growth may be observed in the anaerobic zone of a plate or as bubbles in fluid media. Now let's talk about specimen collection and transport. Sites with heavy resident flora, such as the oral cavity, gastrointestinal tract, or genitourinary tract, are not suitable for anaerobic culture. The best method is to aspirate material with a syringe and needle, and then place it into a special transport vial or tube under reduced conditions. Swabs are not preferred because they do not maintain anaerobes well. Moving to culture techniques. The classic method is the Gas Pak jar technique. A catalyst, usually palladium pellets, is placed in the jar, and a special envelope generates hydrogen and carbon dioxide when water is added. Indicators such as methylene blue or resazurins signal whether oxygen is present. Methylene blue turns blue and resazurin turns pink when oxidized, but both appear clear when reduced. Other methods include anaerobic bags, which are clear bags containing gas-generating ampules. Plates can be read directly without opening the bag. The roll tube technique, which uses pre-reduced anaerobically sterilized media, inoculated under a constant flow of oxygen-free gas. The anaerobic chamber, where plates are transferred through a passbox into the chamber. Inside, an incubator and palladium catalyst maintain the anaerobic conditions. Now for the examination of primary plates. Some characteristic colony morphologies can point to specific organisms. Pitting colonies suggest Bacteroides ureolyticus, though Eikenella can look similar. Large colonies with a double zone of hemolysis suggest Clostridium perfringens. These should be further tested on egg yolk, agar for the Nagler reaction. Breadcrumb or speckled colonies, often with slender gram-negative fusiform rods, suggest Fusobacterium nucleatum. Molar tooth colonies, formed by gram-positive branching rods, suggest Actinomyces. On BBE agar, dark colonies greater than 1 mm indicate Bacteroides fragilis. On KVLB agar, pigment should be examined, and colonies may fluoresce under UV light. Finally, an aerotolerance test is performed on each colony type. Colonies are subcultured onto an anaerobic blood agar plate and onto a chocolate agar plate. The chocolate agar is incubated at 36 degrees Celsius in 5 to 10% carbon dioxide, while the anaerobic plate is incubated in a gas pack jar or chamber. If growth occurs on both plates, the organism is facultative. If it grows only on the anaerobic plate, it is a true anaerobe. This chart focuses on differentiating gram-negative anaerobes using antibiotic susceptibility discs, biochemical reactions, and colony characteristics. Let's begin with Bacteroides fragilis. It is resistant to vancomycin, resistant to kanamycin, and resistant to colistin. It shows variable results with indole, is negative for lipase, positive for escalin, and negative for hydrogen sulfide. On BBE agar, it produces black colonies due to catalase activity, making it distinctive. Porphyromonas acrolithica is sensitive to vancomycin but resistant to both kanamycin and colistin. It is indole positive, lipase positive, but escalin negative and hydrogen sulfide negative. Colonies may fluoresce brick red and sometimes produce black pigment. Prevotella intermedia is resistant to vancomycin, resistant to kanamycin, and resistant to colistin. It is indole positive, lipase positive, escalin negative, and hydrogen sulfide negative. Colonies may also fluoresce brick red and may produce black pigment. Bacteroides ureolyticus is resistant to vancomycin, resistant to kanamycin, and sensitive to colistin. It is indole negative, lipase negative, esculin negative, and hydrogen sulfide negative. On plates, it shows pitting colonies and is strongly urese positive. Fusobacterium nucleatum is resistant to vancomycin, sensitive to kanamycin, and sensitive to colistin. It is indole positive, lipase negative, esculin negative, and hydrogen sulfide negative. Microscopically, it appears as thin fusiform rods with speckled colonies. Fusobacterium necrophorum is resistant to vancomycin, sensitive to kanamycin, and sensitive to colistin. It is indole positive, lipase positive, esculin negative, and hydrogen sulfide negative. Its rods vary in length and width. Finally, Fusobacterium mortiferum is resistant to vancomycin, sensitive to kanamycin, and sensitive to colistin. It is variable with indole, positive for esculin, and positive for hydrogen sulfide. Morphologically, it presents as highly pleomorphic rods. The antibiotic patterns, vancomycin, kanamycin, and colistin, along with indole, lipase, esculin, and hydrogen sulfide reactions are essential in distinguishing these gram-negative anaerobes in the lab. Let's focus on differentiating gram-positive anaerobes, which are important to recognize in the clinical microbiology lab. We begin with Clostridium difficile. This organism is well known for causing pseudomembranous colitis, or PMC. It is usually identified with immunodiagnostic tests rather than culture because of safety concerns and time. Emerging resistant strains are increasingly reported. On CCFA agar, colonies have a distinct horse-stable odor. Clostridium difficile is also a spore former, making it highly resilient in the environment. Next is Clostridium perfringens. This species is recognized by its double zone of hemolysis on blood agar. It produces lecithinase, which can be detected on egg yolk agar, and it is strongly associated with gas gangrene. Unlike some other Clostridium, spores are seldom observed in this species. Clostridium tetani is identified by its terminal spores, which give the organism a distinctive racket-shaped appearance under the microscope. This is the causative agent of tetanus, characterized by severe muscle spasms. Actinomyces israelii is another gram-positive anaerobe. It produces characteristic molar tooth colonies. Microscopically, it appears as branching gram-positive rods and is linked to lumpy jaw infections. Another hallmark feature is the presence of sulfur granules in draining lesions. Finally, Peptostreptococcus anaerobeus, an anaerobic gram-positive coccus, is notable for being sensitive to SPS, or sodium polyanethol sulfonate. This helps differentiate it from other anaerobic cocci.